Welcome to Careers in Discovery, where you'll meet scientists who've forged outstanding careers in biotech and hear about what they've learned along the way. This podcast is brought to you by Singular, building brilliant biotechs. Roland Burli is the Vice President of Drug Discovery at Cerevance, where he and his team are using a unique discovery platform to develop new treatments for brain diseases. Roland joined us to discuss his varied career to date, from landing in LA with just a backpack to working for some of the industry's biggest names. I am delighted to be joined this week by Roland Burli of Cerevance. Roland, welcome to Careers in Discovery.
Thank you very much. Thanks, Tom, for inviting me.
Fantastic to see you. So Roland, let's start with Cerevance. I'm really interested in the work that you are doing in neuroscience and with your NetSeq platform. Tell us a little bit about everything that's going on over there and what you're up to.
Okay, yeah, there is a lot going on. Yeah, so Cerevance is quite a small biotech. It's about six years old or so, based in Cambridge in the UK.
Mm-hmm.
There are people in the US as well, mainly clinical and some senior management, but the operation is preclinical operation certainly is all based here in the UK.
Yes.
The company is really the unique part about the company is a target identification platform, which we call NetSeq. And what's unique about it is so I should say that we're only interested in neurological and neurodegenerative diseases,
Mm-hmm.
all diseases of the brain, like Alzheimer's, Parkinson's, and other grievous diseases. And what we're doing is we're not building hypotheses based on animal data. We're really going directly into the human brain. We're trying to understand the human brain and how the disease starts there. And to do that, we have thousands of brain samples, and basically brain anatomists, they dissect the areas in those human brains, post-mortem brains, the areas where the disease starts. We then take those tissues, we sort those cells into many different cell types, 60 different cell types, and we do deep transcriptomics on all those sorted cells. So because they're pooled cells, we can go very deep. We look at 12,000 genes and transcripts. So we see a very comprehensive picture of how the disease starts, in which cell type it starts, in which region it starts, how it evolves during the disease, because not everybody dies at the very end of Alzheimer's.
No, of course.
The reasons as well. So we can see the progression as well. And we look at age-matched controls as well, because in the aging human brain, the gene expression is changing as well. And we compare that, how this relates to the disease. So you can imagine, with a lot of data about different cell types, many genes in different diseases, different ages and stages, you have an enormous data set.
Yes.
And that's when the selection starts. What are we working on? So we then really, what we're interested in are ideally genes or proteins that derive from these genes, which are selectively expressed in only one cell type in the brain, because that will be better probably for the side effect profile, but also easier for us to understand the pathway and the biology in that particular cell type. It should be dysregulated in the disease, so then you know that you have to up or down regulate it as a potential therapy, and clearly, ideally relatively little peripheral expression as well, because you don't want to have a lot of a peripheral burden and impact upon modulation. And then really, so that's one part. So we select these targets. They do exist. They are definitely, a lot of them are not very well investigated. So there's a lot of new ground to cover, and new genes to look at. Of course, one big aspect is also druggability. We obviously can only work on genes that where the proteins are druggable, and internally we only work on small molecules, although this platform is agnostic of the modality. So in principle, one could access or try to interfere with those targets as well with other modalities, but that's not what we're doing in-house.
Yeah, interesting. And so it's interesting the focus on where these diseases start from, because I suppose, like any type of degenerative disease, it's different from other conditions in that it's not that you're healthy one day and you're sick the next, right? There's a progression of these things, and I suppose it's hard to identify sometimes when and where they start, and that's what you're focusing on from the sounds of it.
Yeah, and it's often not just one isolated gene, but it's a whole pathway which is kind of dysregulated as the disease progresses. So you really want to form a picture and a hypothesis, and not just say, I like this gene, So it has to make sense, but it only makes sense if you actually look at the whole data set. It doesn't really make sense if you just look at one cell type and one gene or so. So that's the power of the platform, I would say.
Yeah, of course. No, it's really interesting, and I think there's very little treatment for these diseases at the moment, isn't there? And of course, there's complexity around that. But if we can, there's the direct result, I suppose, of the work that you're doing, that hopefully we can develop some therapies for them. But also gaining that understanding of them hopefully provides a foundation for other ways to look at these problems as well.
Absolutely, yeah. No, it's it's a very gratifying task, because it's a huge problem in society, and like diseases like Alzheimer's or Parkinson's, almost everybody knows somebody who has that, and to be able to work on this is really a privilege, actually.
No, absolutely. And we'll talk a little bit about how you ended up getting involved in this as we go along, but tell us about today your role is as Vice President of Drug Discovery. Tell us a bit, Roland, about your days. Where do you spend your time? What is it that you're doing in your position there?
Yeah, so I really get involved when we select a target, when we say, okay, this is druggable, there is a good hypothesis, but maybe underexplored, but we need some molecules that we can test the hypothesis and maybe even advance and go towards the clinic. So that's where my team is very involved, or becomes very involved. And then you need, often there is nothing out there. So we have case studies where we basically started with a high throughput screen to find modulators, inhibitors, activators. And then we take those initial hits. We typically we have to improve the potency, the selectivity, the pharmacokinetic properties, so that it's well behaved in vivo, it's orally bioavailable, brain permeable, not too many side effects, drug interactions, and so on. And when all these things line up, then you can start to really profile in advanced disease models, in toxicology safety studies, select a candidate for further development and go into the clinic. And we've done that with 2 molecules that are now in late stage preclinical development, and that's very exciting, of course. So we have projects at all stages. We have very early targets. We have sort of, we call it hit to lead, lead optimization, but then also the preclinical development. And we do have 2 compounds in the clinic as well, which is a whole other story, but that's just to visualize how much is going on at Cerevance. So we're building a whole clinical team as well to progress those molecules.
Yeah, interesting. And all based around this same platform initially.
Yeah, and perhaps what's important to share is, maybe it's obvious, but this is a very multidisciplinary piece of work. So we have chemists, we have people who design a lot, we have people who do set up assays, in vitro assays, we have people then on the neuro side, people who validate those targets in very neuro bespoke assays. That could be electrophysiology, and other assays. We have DMPK, and we do a lot external as well.
Yes.
I mean, we can't do everything internal. So, for example, synthesis and everything that's all done externally. All the pharmacokinetic studies and in vitro studies, ADME studies, they're also done externally. So there is a lot of coordination as well going on. And people, certainly in my team, they're very quickly getting used to work and get something done with the help of external partners. That's very important.
Yeah, absolutely. I think that's the way the world works these days, isn't it? Is you sort of focus on what's your real expertise, and then there are other people who can do the other parts of it, you know, more effectively, more quickly, with greater use of resource, those sorts of things. But that coordination, and we saw this in real highlight during COVID, right? And in the development of vaccines, bringing those expertises together, to take everything further. So it's interesting. I want to go right back to the beginning, Roland. So I'm sure we'll come back to Cerevance, but you, if I understand correctly, you're a chemist originally.
Yes. I studied chemistry and did my PhD at the ETH in Zurich, which was a great time. You know, it's really my sort of, I would say, first big step in my career was to learn how to make complicated molecules and develop methods to do that and so on. So I had a great time doing that and really enjoyed that. And then sometimes you have certain plans in your life, but it always comes different. So that's funny. So I then, after my PhD, I went to Caltech for a postdoc. And that was really sort of the early beginning of, I would say, another phase in my career, because at Caltech I did a more bioorganic postdoc. We designed molecules that bind very sequence specifically to DNA. So that was sort of the first time I really got involved in analyzing crystal structures, trying to understand how molecules interact with each other. Like small ligands with a larger molecule, biomolecule. In that case, it was DNA. Later on in industry, obviously became proteins. That's what we often mostly do in industry. But that sort of was a new fascination, I would say, rather than just making a molecule, but also then trying to understand what function does it have in biology, and how could it help, essentially.
And that was around about the late 90s that you headed out there. Which must have been a fascinating time to be in this kind of work in California, because there was so much interesting science and so many cutting edge companies and research departments around.
So originally, before I left, I was sure I would immediately go back to Switzerland and work in Basel. That was not uncommon. But in the late 90s, as you say, the West Coast was very vibrant. I ended up joining a small biotech in San Francisco. Really enjoyed that. I was working mainly on antibacterial projects. That was then the first time I also worked on proteins, not just DNA. Very, very interesting time. I've learned a lot there. It was a shame that when 9/11 happened, things got a bit harder. That's unusual that does happen, but it made me rethink, and partially also for private reasons, I basically went back to Los Angeles, and I was able to join Amgen, which was in around 2003, which was really up and coming. There was a big expansion in small molecule drug discovery, paired up with the protein work that Amgen was certainly very famous for at the time. So I had a great time there. Had a really nice team, worked in many indications and across therapeutic areas. So there was a big learning curve as well there. So absolutely.
Yeah, certainly. And I'm always interested in kind of the root causes of people's careers, or the origins of people's careers. I mean, for you, Roland, why why science, why chemistry, why drug development? You know, why this career originally for you?
Yeah, it's a good question. I mean, I was always interested in science, but I didn't really know what. It could be physics, it could be astronomy, chemistry, math. I mean, I biology. I was I couldn't say that I was always sort of drawn to chemistry. I think I just enjoyed, I guess, understanding how things work, how nature works. But whether that's like looking at the stars or into a textbook, that's I wasn't really much difference to me. And then it was almost a bit of a coincidence. I mean, we had a really good teacher in high school in chemistry, and he kind of made it look very attractive, and I don't regret that, honestly. I mean, I felt very comfortable during the studies. I really enjoyed the studies and wanted to specialize more and did a PhD, and that one thing led to another, So it's not always all planned out. It's sometimes you just do something and then you take a risk as well. I mean, going to the US to complete it. I mean, I arrived in Los Angeles with a backpack, nothing else, and stayed there for 11 years in total. So sometimes it does come different than you expect or you plan, but that's okay. I think that's
absolutely
probably one message as well. Just stay a bit open-minded and do where you feel like what's best.
Yeah, no, certainly, and I think serendipity plays a big part in these things, right? And I think a lot of people get into science because they find it interesting, and then they follow their interests, right? And they follow the things that intrigue them.
Yeah.
I guess this might be a slightly more difficult question, so take a minute if you need to. But having worked in drug discovery now for several years, it can be a pretty tough gig, right? A lot of things fail, and a lot of things that you're very excited about don't work out, and every now and again there's a little breakthrough, but a lot of it is hard work, and you got to be resilient to build a career in drug discovery. I think what I often come across is you also have to remain passionate and enthusiastic, and things like that. So what is it that keeps you engaged with it now at this point in your career? You were pursuing your interests, and I'm sure that's still part of it, but what do you think keeps you going?
Well, I think it's on several levels. I mean, you're absolutely right. The resilience and the tolerance of failure or so-called failure has to be very high in this business. And because most things that we do ultimately fail at one point or another.
Yes.
But I don't necessarily see it as a complete failure, because every failure teaches us what not to do. And you can still, sometimes you publish these things and then others don't make the same mistake. And maybe it's not you making the final drug, but maybe it's somebody else who used your data to make the final drug. And for me, the big picture is in this industry that we want to be successful and we want to help patients, and that's really what matters. Of course, you have the ambition that I want to be the one to make the drug, or I want my company to be able to sell the drug and so on. That's only natural. But the bigger picture is absolutely that as a community, we want to be successful because we want to help patients. And I mean, that's one aspect. The other aspect is often not always just you're unsuccessful until you sell a drug. I mean, sometimes having a good hypothesis and saying, okay, I've designed this molecule and suddenly it's 50-fold more potent or so. Those are big successes that you can absolutely celebrate, and you should celebrate. Or you nominate a preclinical candidate or something like this. You have a molecule that is absolutely ready to go into the clinic. Those are massive successes that absolutely you should celebrate and be proud of. So that there are many steps to a final goal, and each step is is rewarding. So I totally see that. And then there are other rewards as well. You know, as you're longer in the industry and maybe you have a group and you see how the people in your group evolve, develop, become more experienced. And I mean, that's big reward as well. If you see, it's like, well, you know, I hired this guy and now they do this and that and the other. I mean, that's, there are many ways of having successes in this business, I would say. So it's a fascinating industry, but you're right. It's, you don't go home every day and say, okay, I can see I built this chair or something. You don't know a physical product, of course.
Yeah, of course. Or, you know, as you say, though, I suppose and I guess this is true for any kind of scientific research-based career, but certainly in drug discovery, there's a unique element to this in that even if your program doesn't work, you know, maybe at least you get a paper out of it, or you contribute to the sort of general knowledge pool, and then maybe a few years away on the other side of the world, someone else sees that and does something with it. And I guess it's always, as you say, looking at that big picture of, okay, what we were trying to do wasn't quite the right thing, or it didn't quite work, but maybe this contributes to this thing over here that I don't even know about. And I guess that's kind of one of the pretty unique things about this kind of work.
Absolutely. And I mean, just maybe a word about that is publishing, I think, does remain very important. I mean, if people join this industry, I would definitely not recommend to stop publishing, because it's good for your personal development, but it's also important for the company. And as we just said, you know, sometimes others pick something up and drive it forward into a different direction. So we can't always publish everything in sort of real time. Sometimes there is a delay, but I'm definitely an advocate of still doing that, not just in academia.
Yeah, absolutely. So picking back up on your career, so GeneSoft, Amgen was a relatively small company when you joined, but was on a big growth drive. You then headed out to Biofocus and eventually found your way to MedImmune, and that then became AstraZeneca. So there's a lot of differences in scale in some of these companies. And tell us a bit about that. So, you know, your career in small biotech, your career in much, much larger companies. Tell us a bit about that journey for you.
Yeah, so, I mean, Amgen was pretty big when I joined.
Okay.
And it was definitely way different from a small biotech. And it was a very different learning curve, you know, because for in a small company, sometimes you have to build up things, you have many more little functions or so, and you have to be creative with how you get things done. In a larger company like Amgen or later on AstraZeneca, that's a bit different. You have almost anything you want to do. There is somebody who did a PhD on that, left or right. So the bigger challenge is more to find that person. But that's definitely a different wealth of resources. That's for sure. But sometimes it's harder. Just because the resources there doesn't mean you can use it. So you know, there are other obstacles and decision makings which are perhaps a bit easier in a small organization. So every organization has its unique pluses, I would say, certainly. But yeah, for us, I mean, we, you know, for very private reasons, we relocated to the UK. I didn't necessarily want to leave Amgen at the time. I mean, that was really a private decision. But when we moved here, I was lucky enough to have had a few options, but I decided to join a emerging CRO, Biofocus, at the time. So I felt we did a little bit of outsourcing at Amgen at the time, but I didn't really think I understand how to use this. So I thought this was actually, I consider this an important part of my career. Joining a CRO was really interesting because I really learned what are sort of the the pros and cons, what are the pressure points, and how can you use a CRO in the best way to really advance your own research. And I think now this is very common. Most organizations outsource some activities at least, and it's been a very fruitful experience for me, and I enjoyed that time, certainly.
Yeah,
so a lot of different projects and organizations, had insights into different organizations, how they operate. But then, I did join the neuroscience group at AstraZeneca after 5 years at BioFocus, because I felt that was kind of a logical extension, because that was sort of a semi-virtual organization that was being built up at AZ in small and large molecules, antibodies and small molecules. And we did outsource a lot there, and basically ran projects from AstraZeneca, but similar to here now, we did synthesis and a lot of DMPK and testing. We did that all external. So it was good to know how things work in the outsourcing business.
Yeah. And what are the things that you feel you really picked up about that? Because I think, you're right. It's at a fundamental level, the work is somewhat similar, right? Mechanically, there's a lot of similarity. But as you say, the pressure points are different, and the business model is different, and the commercial realities of the business are different. So what were your observations from that time at BioFocus that you took with you?
Well, I guess there are many. I mean, as you say, how does it work on the business side? I mean, purely internally, you don't really need to worry about that too much. But when you start to negotiate contracts and think about how does that part work, then you do need to worry. You need to also build an element of trust, I would say. It's one thing to have somebody in your team that runs an assay, but it's something whole entirely different if you have to call somebody up in Shanghai and say, can you run this assay, and you trust the results they're doing. You've never seen these people, you don't know how they work, and you kind of have to almost virtually try to understand how does it work in that lab, and do I trust those results? Or I mean, whether it's China or local or so, it doesn't matter. If you don't, you typically don't know the people, and you still need to interact effectively. So all of these things, I would say, I've learned quite a bit in that respect. There are probably areas where it's harder to outsource and areas where it's easier. But it's a good experience.
Yeah. And you talked earlier about the leadership element of your career as it's progressed, and kind of seeing people's careers develop and taking real satisfaction and pride in that. As you've moved into these leadership roles, everybody approaches them differently, right? And it's got to come from who you are and what you're about. But what are your thoughts as a leader on kind of your approach to it and the things that are important to everyone? I think has sort of leadership principles, right? And what would you say about yours?
I would always like to surround myself with the best possible people.
Sure.
And the biggest success is of course you have a core area where you grow and you feel strong, and then you have other areas where maybe you're responsible but you're not so strong yourself. And particularly there, you need strong people around you, but everywhere. I think that's the most important thing, to have people who are good scientists and who are team workers, team players. That's really important. And then once you're there, then the question as a leader is really, how can I help these people to really do the job even better, even more efficient? How can I help them grow for their own sake, but also for the company's sake? Because if they grow, they produce more. So but it's a win-win situation, and it's fun if people are in such a stimulating environment. I am not a big fan of like, well, I'm the boss, I know it best, and others have to follow hierarchically. So I think everybody have their own core expertise, and when it comes about solving a certain problem, then it doesn't matter what your title is. What matters is the one who has the best idea.
Yeah.
And I could be the person who's new on the block, or I could be somebody who's been in the industry for whatever 20 years. My job is perhaps then to evaluate and in some cases make a decision, but the best ideas, of course, don't always come from me. That's for sure. I mean, that's the principle, I would say, by which I operate.
Yeah, I suppose particularly in, as you mentioned right at the beginning, a very multidisciplinary environment, then you aren't going to know as much as some of the people in your team, right? About other things, because that's why they're there. They're the experts.
Yeah, and another good thing is you don't need to think that you need to solve all the problems yourself. I mean, that's why you have a team, and that's even if the team is maybe too stretched or doesn't have the internal expertise to solve things, then you go external. There are consultants, there are CROs. And I mean, I feel like a part of my job is also to maintain and that network. Where can I tap into if I'm running dry, if I don't know how to solve a problem? Who can I ask? Who could I pay to give me an advice? Consultants or so. I mean, this is all very normal in the industry, but maybe not so normal when you freshly come out of a PhD or a postdoc. You're very used to, I need to do my own project, and that has to go away. You once you're part of that team, then you really have to reach out, and the goal is to get it done as quickly as possible and with a good quality, of course.
I think there's an interesting bit of deprogramming that needs to be done there, right? Over the first few years of someone's career, that you sort of go through school and your success is based on knowing the answer.
And some people do pick it up quickly, and others it takes a bit of time.
Yeah, no, it's interesting. We could get into a whole debate about that, so so I guess, Roland, I'm really interested. You've probably touched on some of these as we've talked, but thinking about your career and the sort of transitions in your career, and we've talked about some of the things that you've learned. If you were talking to someone who was perhaps early in their career, or perhaps someone who just finished their PhD or was in their first role in industry, or was earlier on, or maybe if you were reflecting back on your time then, what do you think are the key things you've learned that you'd advise them to think about, and the key pieces of advice you'd give to someone earlier on?
I would say the first few years, I would definitely, what's really important is to establish yourself, and that mean by that, I mean, you learn a certain skill set in academia, and pretty much if I think back, for me, it was synthetic chemistry. And then the next thing is to really apply that in the context of a drug discovery project. And then you really have to learn, you have to learn about the basics in other disciplines, like pharmacokinetics, pharmacodynamics, in vitro pharmacology. There is a lot to learn, and I would say that takes a few years to really get to understand the basics there. And I would take my time to do that. And there are many ways to do this. You have colleagues that you can learn from. You have courses that you can attend nowadays. I mean, with you can find a lot even online, but there are good conferences as well, and I would definitely encourage people to attend that, to do that. And then, of course, that apply it, work hard and make sure that you can publish a bit as well, so you have a proven track record that people can also see, and then good things will happen. That's very often the case. There are a lot of good people are needed in this industry, so I think I would focus on really establish myself as a good scientist and learn the business from the ground, and then the next steps they come. You just have to be open-minded, you know.
Yeah.
I personally, I did enjoy the geographic changes. I felt I learned quite a bit working in, or you know, coming from a very Swiss background.
Yes.
And then having spent quite a good amount of years at the West Coast and now in England. I mean, the cultures are different. The way people approach science problems and so on are different. But you learn wherever you go. You learn a lot.
No, absolutely.
That change can certainly be enriching.
Yeah. And how different was California to Switzerland? Do you remember?
Very different. The weather was better. No, it was culturally, it's very different, I would say. For me, the major step was I'm talking now, I can't say how it is in Switzerland today, but you know, back in the late 90s, if you have an idea in Switzerland, it's often like that will be pressure tested from all sides, before somebody says, well, yeah, it's a good idea. Whereas if you go to California, it's probably the other extreme, where people first say, oh, it's a great idea, and then they start to think about whether it's a good idea. You know? So, but I mean, if you think back historically, a lot of big inventions did come from the West Coast. And I don't think that's such a coincidence, because they have this open mind to, I mean, no idea can be crazy enough. The crazier, the better. And then many things fail, but failure is not necessarily, doesn't lead to a complete downhill spiral. I mean, these people, what's assumed is that, okay, if somebody failed, then they won't make the same mistake twice. So they're more experienced. And that's a very interesting positive attitude, I would say. So
I think so.
Yeah. Sorry.
And I was going to say, I think sometimes we don't realize how much the culture we grew up in influences how we think about things and how we approach things. And I remember reading about a study that someone, I can't remember who it was, but someone had done years and years ago, and they'd taken groups of people of all different backgrounds, but grouped by nationality, and they got them to do a project. And I think the project was to build a bridge in a battleground or something like that. And they wanted to see the cultural differences. And you know, the Americans were at one extreme of things, where they spent, if they had 10 hours, they spent 15 minutes planning and then just started doing it and made loads of mistakes, but they eventually got there because they kept correcting as they went along, and they just, you know, they leapt into action before anyone else. Whereas the other extreme was the Japanese. And the Japanese, if they had 10 hours, spent 9.5 hours planning the project and then flawlessly executed it. And they all arrived at the same point and roughly at the same time, but they did it in very, very different ways. And I think it's interesting to kind of, you don't notice the things that are really fundamental in your own thinking, right? And that's where getting out into the world, I think, probably helps people.
Yeah, absolutely. Yeah. And then you have to find your own style, what works best for yourself.
True. And speaking of what works best and what's next and your own style and things like that. So tell us about, so Cerevance, you've said, you've got some things in the clinic, you've got some things in late preclinical, there's lots of pipeline there as well. We're heading, as we record this, it'll be released next year, but as we record this, it's towards the end of 2022. Tell us about 2023 at Cerevance and where you're headed and what's next.
Yeah, so I mean, when I joined, even like 4 years ago or so, we really had an early pipeline by and large. You know, we started projects, and now they go forward, and you know, all being good, they will go into the clinic in next year and the year after, and new projects are being generated, but the company as a whole is definitely changing enormously, because it's a big deal to have clinical stage and preclinical projects. I mean, we had this year, we were fortunate to be able to report that we had a a positive result in phase 2 in Parkinson's disease with the lead project. And that goes forward, that continues to go forward. Then we have a phase 1 project, which looks promising. And the others, if one or two more projects or compounds can go into the clinic, then we really have a lot to do there. So we have to build up an entire clinical team. So
yes.
we recently hired a chief medical officer, and that's absolutely exciting for us. So that'll be a very different company from the outside, and clearly it validates the platform, it validates the whole target identification, the way we do work, because we do it differently. We dare to be quite novel here compared to other organizations, so that needs to be validated. But we're making huge steps towards that, and that's definitely exciting. So
it's really encouraging to hear, because I think this is a problem that's been with people for centuries, right? And it's incredibly complex, and that's meant that there's very little there are very little options for people when they encounter these conditions. But clearly approaching it in the novel way that you are is yielding some results. So it's really exciting to hear, and of course we wish you the best of luck with it, Roland.
Thanks. No, that's good.
Thank you so much for joining us.
Thank you.
Thanks very much for listening. Careers in Discovery is sponsored by Singular, helping you to build a brilliant biotech company. Biotech leaders spend far too much time, money, and energy on hiring and people issues. Head to www.singular-biotech.com to learn how you can recruit and engage your team more effectively, so you can focus on developing medicines, treating patients, and saving lives.